final speaker is Dr. Zachary Simmons. Over 20 years ago, Dr. Simmons and nurse Sue Walsh founded what is now the Penn State ALS Clinic and Research Center, an ALS Association Center of Excellence. The clinic provides ongoing care to over 200 individuals with ALS. Dr. Simmons and his team are well known for their development of an ALS-specific quality of life instrument. And the center helped pioneer the use of telemedicine in ALS for clinical and research purposes. The current research program at Hershey is robust, including clinical treatment trials. Dr. Simmons has authored more than 100 peer-reviewed scientific articles and currently serves as editor-in-chief of Muscle and Nerve. Having known Dr. Simmons for his 20-year plus and counting tenure, he is the first one to raise his hand to volunteer to help and make himself available, whether it's for an ALS conference like this, our Hershey Walk, interpreting research into lay terms, and serving on countless ALS scientific committees, including the platform trial, which is the topic of his remarks. He is a devoted fan of his clinic team, which is reciprocated and is beloved by all who seek his care. Dr. Simmons, your turn.
So hello, I'm Zachary Simmons. I'm a neurologist at Penn State University, and I'm very pleased to be here today. I want to thank the ALS Association Greater Philadelphia chapter for giving me an opportunity to talk. I don't think that this is the type of environment that any of us would have imagined a couple of months ago, but nonetheless, this is a very important topic, and I'm really pleased to have the opportunity to present this to you. I have a few disclosures here of companies that I act as a consultant for or do sponsored research for. And what I wanted to do was start off by giving some background to the ALS platform trial design. So this trial design is the brainchild and the really the result of efforts by the Healy Center at Massachusetts General Hospital. I've put up a photo here of some of the investigators and scientists and physicians who are behind this. And much of what I'm telling you today will be based on material that they've presented earlier. Some of the slides with permission are being used from their presentations as well. The platform trial is supported by a number of organizations, including the ALS Association, and including a number of other nonprofit and advocacy organizations. So why do an ALS platform trial? Well, the purpose of it is really to cut time and cost and to increase the effectiveness of these therapies. So, in the traditional study, a disease is identified, in this case ALS, and a therapy is given, therapy A in this case, and then the outcomes in terms of how people do and how they respond to the drug are measured. It's pretty simple and straightforward. The platform trial design takes that a step further. Still, the disease is identified at the beginning, in this case ALS, but instead of a single intervention, the study involves more than one intervention ongoing simultaneously. In this case, as I'll be talking about today, 3 simultaneous therapies. And the goal of this is to do 2 things. First of all, to reduce the cost, and secondly, to reduce the time involved with being able to come up with effective therapies. So, to give an example of how platform trial design can expedite therapy development, I'd like to present a simple mathematical model. So statistics have been done that show that if you're testing 10 different therapies, and each of them has a 10% chance of being effective, that under the traditional model, one drug, one trial, it could take as long as 12 years and as many as 3,600 participants, 2,400 on placebo on drug and 1,200 on placebo, to come up with one effective therapy. In contrast to that, in the platform trial, the same 10 therapies could be tested by a much lower number of participants and a much smaller number of placebo patients in a 4-year period of time, and yet come up with the same probability of identifying an effective therapy. So overall, much quicker and much less costly, and involving many less participants who have to be on placebo or drug in order to come up with an effective therapy. I'd like to go through the process itself. So the process begins with consent and randomization. The participants that initially provide consent to what we call a master protocol. So those individuals are screened for inclusion and exclusion criteria, and I'll go over that in just a few minutes. And those that are determined to be eligible based on inclusion and exclusion criteria are then randomized to one of 3 therapies. Let me show you a picture of that, which I think will explain it better as I go along. So if you look at the box on the left that says Enrollment Platform Trial, the star that's just to the right of that indicates that people then give consent to be in the study, and they are then screened for inclusion and exclusion criteria, how old they are, how long they've had the disease, and so forth. And if they are successfully clear the inclusion-exclusion criteria, they're then randomized to one of 3 therapies, which here are indicated as therapy A, therapy B, and therapy C. So that's really the first step. And individuals do not have a choice as to which therapy they would go into, but they are randomly assigned to one of the 3 of those. Now, within each of those therapy arms, A, B, and C, participants are then once again randomized to either active treatment or placebo, and that's a 3-to-1 ratio. So again, let me show you a picture, which I think will, it's the same picture, but let me show you a different part of it. If you look at the set of stars that are in the middle of the screen, that's a second consent process. And people in therapy A, for example, are then randomized to either active drug or placebo. People in therapy B, also active drug or placebo, and the same for people in therapy C. Now, individuals will know whether they're on therapy A, therapy B, or therapy C arm, but within that arm, they won't know whether they are randomized to the active drug or placebo. All they'll know, for example, if they're in therapy A arm, is that they have a 3 out of 4 chance of being on therapy A and a 1 out of 4 chance of being on placebo. Same in therapy B, same in therapy C. Overall, there are 160 participants nationally per arm. Because there are 3 arms, there are 480 participants overall. And as I said, they're randomized 3-to-1 drug to placebo, meaning that there are 360 individuals on the drug and 120 on placebo. And there are 2 other pieces of this that I'll talk about in just a few minutes. One of them is, as you can see at the right, there's an open label extension. And then that other big red arrow indicates that people can actually go back and re-enroll again under a limited set of circumstances after they've completed the trial. And I'll go through both of those aspects. So overall, in this trial, there are 54 participating centers nationally. They're shown on a map here, and each center will be enrolling individuals in therapies A, B, and C. So when we look at studies, we look at objectives and we look at measures for how we can determine whether those objectives are achieved. Of course, the objective of any ALS study seems in some respects obvious. The objective is to either slow down the rate of progression of ALS or reverse ALS or cure ALS. So the objectives seem clear. But in order to do these studies and in order for the FDA and other regulatory agencies to really know, and for us as physicians and scientists to know whether a drug is succeeding, we have to pick certain measures and look at them very carefully. So the primary measure of effectiveness in this study is to look at the effectiveness of the drug compared to placebo by measuring the ALS Functional Rating Scale score, the ALS-FRS revised, or ALS-FRS-R. That's a measure of ALS function, and what we'll be looking to see is if a person is on active drug if a group is on active drug compared to placebo, is their rate of progression different? We don't expect that the drugs in this study will cure ALS, unfortunately, but what we're hoping is that they will meaningfully slow the rate of progression of the ALS, deficits that the ALS produces. So meaningfully slow the rate of progression of the ALS functional rating scale. There are other objectives to this study as well, what we call secondary efficacy objectives. And so in addition to measuring the change in the ALS functional rating scale, we'll be looking at the effect of the drug on other measures, measures of respiratory function measured by vital capacity, measures of muscle strength using certain handheld devices. And for those patients who unfortunately pass away during the trial, we'll be looking at survival as well. And all of these, again, are to determine how these change in people who take the drug relative to people who take the placebo. There are also safety objectives. So any study is done with one goal being making certain that the drug is safe. If a drug is effective for ALS but is very, very toxic, it's not a good drug. So we'll be looking at safety by looking at adverse events, side effects in other words, laboratory tests to monitor what it does to people's liver tests, their kidney tests, and so forth, and then cardiograms. So who can enroll in this study? What are what we call the key inclusion criteria? So this study is designed for people with either sporadic or inherited familial forms of ALS, who have a time since onset of weakness of 3 years or less. They have to have a vital capacity of at least 50% of predicted. And I'll just say, by way of explanation, that 50% cutoff is very liberal compared to most clinical trials that I and others have been involved in. Many ALS trials in the past have required 70 or even 80% vital capacity in order for people to qualify. 50% will allow many more people to qualify for this study than have qualified for many past studies. There are statements there about riluzole and about edaravone, which goes by the brand name Radicava. And the easiest way to summarize those two statements are that if a person in a study can be on either of those, or they can be not taking either of those, and that's fine. But what people can't do is to start one of those partway through this clinical trial. So again, they can either be off riluzole or Radicava, they can be on riluzole or Radicava at the beginning, but they can't be off it and then start it partway through the study. The study lasts 24 weeks and consists of a combination of in-person and phone follow-ups. And at the conclusion of the 24-week placebo-controlled period, participants have some choices. They can schedule a 40-day follow-up safety phone call, and they can then say, I'm done. Or they can have the option to receive their study drug in an open label extension phase. Now, as I say here, in the open label extension, the study drug will be provided until the drug is either approved and available or the manufacturer terminates the development of the drug. What we mean by open label extension is that if people are in therapy arm A, then at the end of the study period, even though they don't know and we don't know whether they've been getting the drug or the placebo, they can opt to be in the extension, and then in that extension, they will get the drug, and that's guaranteed. In other words, they will not get placebo. The same for people in therapy arm B, they can be guaranteed to get drug B, and the same for therapy arm C, they can be guaranteed to get drug C. The open label extension phase, again, is to follow them for safety and also for effectiveness. It's a combination of in-person and phone follow-ups. Or instead of going into the open label extension, and instead of opting to just stop involvement, people can say, oh, I'd like to go back into a different arm of the study if it's still enrolling and if they still qualify. What I mean is that if people have been in therapy arm A, they can say, well, I want to try to see if I can be on a different drug. And if they still meet inclusion-exclusion criteria, they then will be randomized to either therapy B or therapy C. Now, if people do that, they need to keep in mind they can also receive placebo in either of those two arms, just like they could at the very beginning. So the only way to guarantee that they'll get drug is to stay in the arm they're in and go into the open label extension. If they want to try something else, they can if they still qualify, but they may receive placebo. So even though we're only trialing 3 drugs, the first 5 have actually been selected for the platform trial. These are the names of some of the manufacturers here, and these 5 were selected from 27 different companies who submitted proposals. The other 2 drugs will likely be trialed, likely be trialed as soon as the first 3 are underway. What I wanted to do here was to go over the 3 drugs that are currently being used in the study that's about to open. In no particular order, one of the drugs is called zilucoplan. Zilucoplan is a complement inhibitor, and complement is part of the immune system. So this drug acts on the immune system, and interestingly enough, it's undergoing trials for autoimmune diseases. ALS is not obviously autoimmune, but there is inflammation within the brain and spinal cord in ALS, and we think this is what zilucoplan acts on. Interestingly enough, this whole cascade of complement activation also occurs with other neurodegenerative disorders, Alzheimer's, Parkinson's, and Huntington's. So this drug may have broader use as well. It's given subcutaneous injection, meaning an injection under the skin similar to what one does for insulin, and it's given once daily. The second of the 2 is verdiperstat, and verdiperstat is what's called a myeloperoxidase, or MPO inhibitor. And myeloperoxidase is an enzyme that appears to be involved in some processes that involve inflammation and oxidative stress. And by inhibiting this enzyme, it's been shown that verdiperstat may slow progression in neurodegenerative disorders. It's being trialed in Parkinson's disease, multiple system atrophy, as well as ALS. And it's a pill, it's given twice daily. The third of the 3 drugs, which I've listed here as CMAU8, is very interesting in the sense that AU is the chemical abbreviation for gold. And CMAU8 is a suspension of tiny little gold nanocrystals. I mean, far beyond the ability to see what's in the liquid. This would look like a clear liquid to anybody drinking it. But it has activities in pathways that involve neuroprotection from oxidative and exocytotic toxic insults, meaning it acts on what we call oxidative stress and excitotoxicity. This is an oral solution. It's given at one of 2 different doses once daily. So people in this arm either get low dose or high dose, or they get placebo. What I wanted to put up here were some platform trial milestones, just to give everybody an idea of how far this has come in such a short period of time, despite the recent COVID epidemic and despite the challenges of starting a new kind of clinical trial. The whole impetus for this began in late 2018, early 2019. And as you can see here in the middle of the table, by September 2019, the 5 candidate drugs had already been selected. Also by that month, the 54 NEALS sites that are involved in this study had been selected. And recently in March, we had a virtual meeting of all the site investigators and the research coordinators as a kickoff for the study. And around the same time, FDA approval was received and IRB approval was received. So this study has moved quite far in quite a short period of time. Now, no doubt, this has been impacted by COVID-19. And I just wanted to touch on that briefly. Almost every center, and literally every center, is now experiencing some disruption to normal workflow. And this affects all research, not just ALS research and not just clinical research, but animal and laboratory research and other disease research. There's been interruptions to workflow, ability to start new studies, enrolling of new participants. That varies tremendously from region to region. And it's also in flux. Federal, state, and local regulations are evolving, and clinical trial guidelines from the FDA, the National Institutes of Health, and institutional review boards are evolving as well, because it's really the goal of all these regulatory agencies to figure out How do we allow these studies to continue in an effective but safe manner in view of the COVID restrictions? So we, meaning the investigators and the leaders of this study, are exploring all possible options for patient enrollment. Remember that accurate and complete uh, acquisition of data is really critical to both protecting the safety of participants and, of course, to assessing the effectiveness of the drug. Unless we can collect data accurately and unless we can collect it regularly, we can't tell if the drug works, and we can't tell if people are being kept safe, if the drug is having bad side effects. So we're all working closely with one another and with our partners in industry and with all the regulatory agencies to look at all options for doing this. So there are many options. And I think that what we'll see is that clinical trials moving forward are in some respects going to be different than clinical trials were, just as the world in many respects is changing as a result of many of the coronavirus restrictions. Virtual visits, including informed consent, are being looked at. In other words, can we obtain consent via video conference? Can we do some of the visits via video conference? Can we do collection of safety assessments like blood work and cardiograms by sending a nurse into people's homes? Can we collect study medication in a different way? Can people send them back in mailers? And can we send them to people's homes in mailers or other prepaid packages? And how do we collect outcome measures? How do you collect breathing measurements? How do you collect other measurements if people can't come in? Are there ways to get those measures collected at home? I think the answer is that there are some. We need to be creative in figuring out how to do that, knowing that we can't collect all the measures remotely that we can in person, but yet knowing that we have to meet a certain bar in order to prove that a drug is safe and effective. So in summary, I'd like to close with a few major points. First is that platform trials can greatly accelerate the path to effective treatment for ALS. As per the mathematical model I showed you earlier, fewer participants over a shorter period of time at lower cost should be able to come up with the same results or equivalent results. Second point is that there is strong support for the platform approach amongst many stakeholders. Platform design is not unique to ALS. This is the first time it's being used in ALS. But the platform trial design is being looked at in cancer and in other diseases as well. And I think will become more and more widely accepted as its advantages become more widely appreciated. The third point is that this is a perpetual trial and will continue to test more interventions until cures are found for all people with ALS. And I do want to close with this because it's an important and I think justifiably optimistic note. Platform trials are perpetual. When one drug is done being tested and drops out of the platform, it either is then put back into the platform so that everybody receives it because it was found to be effective, or it's dropped out of the platform because it was not effective, but other drugs are continuously coming into the platform, so that as additional people are diagnosed with ALS, new drugs are available for them to try as part of the platform trial, even though drugs that had previously been used, their trials may have been filled by the individuals who had ALS previously. The goal for a trial like the platform trial is to keep trying one drug after another, adding back in drugs that work, subtracting drugs that don't, and eventually finding effective treatments and hopefully a cure for ALS. So in closing, I just want to thank the ALS Association Greater Philadelphia chapter for inviting me to do this, and thank those of you who are listening.